My group, as Carol said, is joint between the Department of Chemistry and the Department of Chemical and Biochemical Engineering. And we have quite a mix of students in chemistry, chemical engineering, biomedical engineering. And what we do is use organic and polymer chemistry to develop new materials. And what I'd like to do here is to just give you two brief examples of how a materials-based approach to therapeutics can offer potential advantages over the more traditional approaches. So what do I mean by the traditional approaches? Well, if you get sick today, chances are you are going to take what we would call a traditional small molecule drug. And so I'm showing a few examples here. Certainly molecules like aspirin and penicillin you've probably heard of. These are a couple of anti-cancer drugs, doxorubicin and Taxol. And these molecules are certainly very useful, but they do suffer from some limitations. Many drug candidates, in fact, suffer from such poor water solubility that they don't even make it to market. And in the case of Taxol, it did make it to market, but it does suffer from this difficulty, and that makes administering this drug really quite a challenge. Also, many small molecule drugs are quite rapidly eliminated from the body. They're excreted and filtered into the urine. And so if you're dealing with a molecule like aspirin or penicillin, then that's not a big deal. You can just take it orally as a pill every few hours so that you have the dose inside your body. But if you go to a more expensive and complex molecule like these anti-cancer drugs that have to be administered intravenously, then this really starts to become a problem. Another major issue with many current drugs is that they have a lack of specificity for their target. So for example, with anti-cancer drugs, they act by being very toxic to tumor cells. Unfortunately, when we administer these drugs, they don't only go to the tumor cells, they go all over throughout our body, and it's their action on our normal cells which leads to many of these bad side effects. Now, as we've had many advances in genomics and proteomics, we're starting to see more and more drugs that are based on DNA and proteins, molecules like that. So you've probably heard of DNA, RNA, or oligonucleotides being used in techniques such as gene therapy or antisense therapy. Protein drugs, some of them have actually been around for quite a while. Insulin is used to treat diabetes, of course, and shown here is a peptide, a follicle-stimulating hormone that's used in fertility treatments. And so these drugs also, like the small molecule drugs, are very useful, but they have many of the same problems as the small molecule drugs, and in addition, other problems in fact. So for example, all of these molecules occur naturally in our body, and so our body has lots of enzymes that are designed to destroy these types of molecules. And so when we inject them into the body or take them orally, they tend to be degraded very rapidly. Also, many of the targets for these drugs are within cells, and these drugs, because they're fairly large and very polar, have difficulty entering cells, and so that becomes then an additional challenge that we need to deal with. So what can we do to improve some of these issues? Well, one option is to take our drug molecules and actually package them up into a drug delivery system and then release them from the drug delivery system once they reach their target. So what kind of improvements can this lead to? Well, if our system here overall is water soluble, then we can certainly improve the solubility of our system. By packaging it up, we can also protect the drug molecules from degradation. It turns out also by going to a larger nano-sized system, as long as we don't go too large, these systems tend to circulate for much longer than the small molecules. And finally, perhaps most importantly, we can actually enhance the specificity for the desired drug target. And there are a number of ways to do this, but I'll just give you one example in the case of anti-cancer drugs. So in normal tissues, the junctions between the cells which line our blood capillaries are really quite tight. And so this means that it's very difficult for large systems such as drug delivery systems to actually exit the vasculature and enter into normal tissue. And when they do, they tend to be rapidly carried away by the lymphatic system. If we look at tumor tissue, on the other hand, the blood vessels tend to be much more leaky. That's partly because the tumors are growing so rapidly. They want to take a lot of molecules in because they're going to process these as food, and also because they're growing so rapidly, the construction is rather shoddy. So what it boils down to is the fact that these larger systems can more easily enter tumor tissue. And when they do, tumor tissue also tends to lack a well-developed lymphatic system, so these molecules or systems are not carried away, and so they accumulate selectively in the tumors, and this is called the enhanced permeation and retention, or the EPR effect. So what kind of drug delivery systems can we use? Well, there are many examples. We can use polymers, which for those of you who aren't familiar with polymers, this is basically many small molecules strung together to form a long chain, and we can attach our drug molecules to these sorts of polymers. We can also use the polymers to form nanoparticles and encapsulate drug molecules inside the nanoparticles. If we tune our polymers properly, we can make it so that they have a water-loving end or a hydrophilic end, and also a hydrophobic end. And when we do this, then they will tend to assemble naturally in solution into structures such as micelles. And going into something which is actually very close to what we have in our bodies, you may know that cell membranes are composed of phospholipids. And if we take these phospholipid membranes without all of the other components of their cells, they tend to be called liposomes. And these have also been investigated for drug delivery. Now, what we're working on in our group is actually a polymeric version of these liposomes. So again, if we take polymers that have both hydrophobic and hydrophilic components, and we tune the ratios of these components properly, we can get them to spontaneously assemble into these structures that we would call polymer vesicles or polymersomes. And so as you can see then, these structures are really mimicking cell membranes. They have other advantages such as increased stability relative to the liposomes because we're dealing with polymeric systems. We can encapsulate water-soluble drugs into these systems because we have a water-filled core, but we can also use the membrane to encapsulate hydrophobic molecules, such as hydrophobic drugs. And depending on how we prepare them, we can actually tune the size. So this is a microscopy image, for example, of some micron-sized vesicles that we made in our lab. We made them this size so that we can see them by microscopy. More specifically, here are a couple of areas that we're looking at. So I mentioned that we can target things to tumor tissues based on this enhanced permeation and retention effect, but we can enhance this further if we actually attach things to the surface which will bind to the cell type that we're after. So certain small molecules can be attached here that will bind selectively to tumor cells. Another aspect that's really important when we develop this kind of system is, well, it's all nice that we can package up a molecule and protect it, but when we get it to its target, we want to be able to release it. And ideally, we want to keep it encapsulated in a controlled manner while it's circulating in our body, but when it reaches its target, release it in a very controlled way. And so in order to do this, we're developing this class of polymers that we call end-capped cascade degradable polymers. And the concept here is that we have a polymer that is very stable in the presence of the end cap, but when we remove this end cap under controlled conditions, then we trigger a depolymerization. In other words, an unzipping of our polymer chain and degradation of the molecules. And we want to be able then to tune this using different end caps under different biological conditions. We have a poster, by the way, on that topic just over there. So the second example that I'd like to give is tissue engineering. And again, if we talk about the traditional approach, as you know right now, if you suffer severe damage or failure of an organ, then typically you'll need to have a transplant. And that's rather unfortunate because often in order to receive a transplant, the donor has to pass away. And so there's a very limited number of donated organs available at any given time and a long waiting list for these organs. And so the concept of tissue engineering is quite different, where instead of starting with a full organ, we would actually simply start with the cells, grow them, seed them onto a polymer scaffold material, and then use this to grow our new tissue, which we could then transplant into the body. Now, of course, as a material scientist, we consider this polymer scaffold to be a really interesting and important component of this system. And there are several requirements for the scaffold. So first, we do want it to be biodegradable so that we wouldn't have to go in later surgically and remove the system. We also want it to favor the attachment of cells, because if cells don't like it, then they're not going to want to grow on it. And also, since the cells are growing rapidly within the scaffold, we need to have this scaffold allowing for the diffusion of nutrients to the cells. And that's the reason that many of these systems are quite porous. And there are different ways to engineer these systems to have this porosity. Now, a lot of work has been done so far with molecules that are called polyesters, and there have been a lot of advancements made, don't get me wrong, but these materials do have some limitations. First of all, these polymers tend to lack functional handles, which means that if we want to attach cell signals or drug molecules to these systems, that becomes quite a challenge chemically. And also, when we hydrolyze polyesters, we tend to get carboxylic acid, which are acidic and can be toxic to the cells under some circumstances. So in collaboration with Kibrit McQuinnan's group in the Department of Chemical Engineering, we're working on exploring this new class of polymers. These are called polyester amides because they have both amide and ester linkages in the backbone. And since we have both of these types of linkages, there are lots of different ways these polymers can be degraded. So amides typically would be cleaved enzymatically, and esters would be cleaved either enzymatically or just through simple hydrolysis if they sit in water long enough. Another nice aspect is that these molecules are composed of diols, amino acids, and dicarboxylic acids. And if we look at biochemical pathways, many of the intermediates can actually be used to prepare these polymers, so that when these are degraded, they're going to be degraded to things that are naturally occurring in our body and will be processed and be non-toxic. Another aspect that we're really interested in is incorporating amino acids that have functional groups on their side chains and using these as functional handles for our materials. So why do we care about having functional handles? I sort of alluded to this, but the ideal is that we want to have a scaffold material that will mimic as closely as possible the natural environments of the cells, which is the extracellular matrix. So as I mentioned, we want it to interact positively with the cells, promote their adhesion, their growth, and differentiation of their functions. And so there are some molecules that we can attach to our scaffolds to achieve this. So for example, peptides of certain sequences can promote the adhesion of cells, and there are also proteins that act as growth factors, which will encourage the proliferation of the cells. So we've made quite a library of polymers so far. This is just a couple of examples. What we're doing is tuning the basic components of these systems, and in doing so we can really tune the properties of these molecules. So for example, as you can see here, some of the polymers are degraded quite slowly and others can be degraded quite rapidly. So we want to be able to say, well, we want something that degrades rapidly, we're going to use this structure, or if we want something that degrades more slowly, we can use a different structure. Where is this all going? Well, Kibrit McQuinnan's group specializes in vascular tissue engineering, and that's our target at the moment. So the idea here is to replace clogged blood vessels, and in doing so to prevent a heart attack, which would result from the occlusion of a blood vessel. And if we look again at the current treatments, there are some polymer grafts, but the problem with these is that their material properties are so different from the natural blood vessels that this starts to cause long-term problems, and often these need to be replaced after several years. We can also graft veins from other parts of our body, but then this requires an additional surgery to go in and get the other veins. And there are also mechanical problems sometimes with these approaches. So really, the ideal is to be able to grow a new coronary artery simply from cells. So where we're at right now is starting to look at the interactions of vascular smooth muscle cells with some of these polymers, and so far they seem to really like these materials. And so the next step will be to fabricate scaffolds which are in the shape of these arteries, and then to grow the arteries in a bioreactor. And we have a poster also on this material, which is also over there. So I think the last slide is just to thank my group. I think this picture is pretty recent. Some of these people you can see around here, and I'd just like to thank our funding sources as well, and you for your attention.